have the opportunity to talk about research and in particular sharks. So we're going to try and share screen again, but I've got a suspicion it might take me a couple of attempts. Let's see what we can do. Let's see if this works. I think we're going to get a mirror image, which will then, when I share screen, I'm going to be looking for confirmation. Olivia, is that good? Okay, great. So today I'm going to give you a very short background of my research career, which was in more detail covered by Olivia right there, and how I came to be working here at the Shed Aquarium. Well, I say here, I'm working from home today. I wish this was my office with a nice sofa in the background, but it's not the case. I'm then going to give a very high level background to the biology, ecology of sharks and the conservation issue. A lot of you may know some of this stuff already. If Pete Sorensen's on this talk today, he definitely knows all of this and may have told some of you this already. But then I'm going to give you a case study into the work we did as part of my postdoc in Jupiter, which is a really good example of how conservation research, active research in the real world can translate into management decisions, and how it's a very nice succinct pathway from the study itself to the management action. And then finally, I'll close by talking briefly about the Shed Aquarium Shark and Ray Research Program and some of the links to University of Minnesota. So in picture form, the background not included in Olivia's introduction is I'm from the UK originally. I grew up in northwest London. The UK is not very big, so it's never too far from the ocean, but opportunities to get to the coast were limited. So my connection to the natural world and eventually the ocean came largely from playing in local ponds and streams. I was fortunate to have a garden, and we had a pond in that garden. So I played in that pond. I played with the frogs, the newts, the fish, and became very fascinated by the natural world. I was also aided by the BBC and David Attenborough and the fantastic shows they put out, and they really fascinated me, and I became very fascinated by sharks. But it wasn't until 2002, when I was an undergraduate at Cardiff University in Wales in the UK, that I had the opportunity to conduct field work around elasmobranchs, and that was at the Bimini Biological Field Station in the Bahamas, which is also known as the Shark Lab locally. This was as part of my undergraduate degree. We had the option to do what they called a year in industry. So I took up that option, and I managed to get an internship at the Bimini Biological Field Station, where I worked with Dr. Samuel Gruber, the late Dr. Samuel Gruber. And it wasn't until that time that I realized that a career in marine science was an option for me. This here is a picture of a large tiger shark we caught in the deep Gulf Stream waters off of the island, but I was primarily working with lemon sharks, which I'll talk about in a little bit, but lots of different species, looking at the nursery areas of sharks in that region. Following the completion of my undergraduate, and I began a PhD, and I went back to the Bimini Biological Field Station to conduct that PhD research, and it was looking at the larger individuals. A lot of the work at the lab was based on juvenile sharks, but we had a longline program that was really underutilized in terms of the data. So I brokered a deal with Dr. Gruber that I'd have access to the historical data and then build on that to conduct an investigation into the shark populations and behavior around the island. During that time, I began my networking journey, going to conferences, and I'd become associated with another British elasmobranch biologist called Dr. Nigel Hussey, who's still based at the University of Windsor. And he had been invited by the Cousteau Society to help with a integrated coastal zone management plan assessment for the Red Sea coast of Sudan. And Dr. Hussey and myself went along as to conduct the elasmobranch element of that coastal zone management plan. And there, to this day, are still the most pristine marine ecosystems I've experienced in my life, and definitely the most abundant shark populations I've ever encountered. So following the completion of my PhD, I undertook a postdoc that was associated with a different department at Cardiff University, but I was physically based in the US in Miami. And we were assessing the movements and migrations, the spatial ecology of large coastal shark species occurring between Boynton Beach to the south and Hobe Sound to the north. So that's an area of the southeast coast of Florida. They're essentially between Fort Lauderdale and about 90 kilometers to the north, or around 60 miles. That stretch, that's where we were tagging and tracking sharks, but these sharks were moving a lot further. And it's from that project that I'm going to give you the case study from a little bit later on. After that, I traveled to Canada, where I spent 4 years working at the Great Lakes Institute, and I was physically based in Windsor, Ontario, which is right across the river from Detroit. But the research component of that position was funded through the Ocean Tracking Network, which is based out of Dalhousie University in Nova Scotia. And it was working up in the high Arctic. So I was very lucky to have the opportunity for 4 years to travel up frequently to the high Arctic, specifically to a place called Resolute Bay, which is on the Northwest Passage. And there, my primary focus, I have a picture here of a Greenland shark that we were tagging. But my primary focus was on Arctic cod, which is the primary forage fish in the Arctic marine ecosystem. It's estimated that around 70 to potentially 90% of all energy transfer from lower to upper trophic levels passes through the biomass of Arctic cod, and their distribution in turn defines the distribution of the kind of larger predators, the sea lions, sorry, not sea lions, seals, polar bears, even beluga, narwhal. All their distribution is dependent on both ice cover, but also the distribution of Arctic cod. So that was a very interesting experience. And then following that stint at Michigan State University, where I worked for a year looking at lake sturgeon movements and migrations around the Huron-Erie corridor. Again, not a shark species focus. It was important for me to diversify my portfolio in terms of research to make myself more employable. And sturgeon were the closest things I could find to sharks in the Great Lakes. I took this position here in Chicago, where I'm based at the Shedd Aquarium, but until recently, the large research focus of our program has been based back in the Bahamas. So I've almost gone full circle back to where my first experiences of field science occurred. So very quickly, I'm going to give you a brief overview of the biology and ecology of sharks. You've probably heard this before. Sharks are very old animals as a group. They originated on the planet around 450 million years ago, not white sharks, of course, and this is just a convenient picture. And that is around 200 million years in advance of the origin of dinosaurs. That's quite a common fact that's thrown around, but one not so common is that that's around 100 million years before the origin of trees on our planet. So they've really been around a very, very long time. And during that time, they've survived all subsequent mass extinction events, which is a really quite a big feat of survival for an animal group. I mean, there are other examples, of course. But during that time, that's afforded them the opportunity to evolve into a very diverse group of animals. Now, I keep saying sharks. I'm talking about elasmobranchs broadly in a sense. Elasmobranchs being sharks, skates, rays, chimerias. You can expand that further to include things like wedgefish, like guitarfish. Sometimes sharks, the term is used as a catch-all for all elasmobranchs. There's around 12 to 1,300 species of identified elasmobranchs currently existing on the planet. Specifically, sharks within that group is around 500 that we know about. We discover around 5 to 10 species, new species per year, typically in the deep ocean. Some of those are found to be genetically differentiated. That's true of some of the hammerhead species, but lots of different species. And as you can see from the picture on the left, a wide variety of morphologies and sizes, which in turn facilitates a wide variety of niches that they are occupying, primarily in the marine ecosystems, but some of them, particularly the rays in the freshwater ecosystems as well. And within those 500 species of sharks specifically, there's some of the more extremes of the animal kingdom. For example, the largest existing fish is the whale shark. It's been measured to be around, the largest individuals measured were around 40 feet in length, but it's estimated that they could quite easily grow up to 60 feet in length. So really, really large animals. Thankfully, they're plankton feeders and not something we have to be worried about. Otherwise, I don't know if I'd be traveling around in the small skiffs I frequent on the ocean if these guys were after larger prey. One of the fastest fish, not the fastest fish in the ocean, I think that accolade goes to the spearfish. I think it's a type of billfish. But one of the fastest fish is the mako shark. The fastest clock fish is the spearfish. It is possible that the mako shark is faster because the mako shark eats the fastest fish in the ocean. So in order to catch it, you would think it might have to have similar, or if not superior speed. They've been clocked at around 40 miles an hour, but they are estimated to be able to exceed 60 miles an hour because when they were clocked, they were following a bait with the camera and the boat itself was going 40 miles an hour. The individuals were dropping back and then accelerating, so they were definitely moving quicker than 40 miles an hour. The exact speed is not possible to be measured in that scenario, or at least wasn't at the time. And if you put that in context, the fastest land animal is the cheetah, which runs at around 60 miles an hour, similar speed estimated for the mako shark. But the mako shark's moving through salt water, which is around 800 times more dense than air, so at sea level. So that's really quite a feat to be able to move that quickly through such a thick medium. It's quite niche, this one, but the longest pregnancy of any known vertebrate is estimated to be the frilled shark. I've heard people say the coelacanth. I think the estimates for the coelacanth are 3 years. But a group from Japan looking at embryo development estimated that frilled shark gestation periods are around 3.5 years, which does eclipse the coelacanth. Now, obviously, this is an estimate, but it's very possible that the longest gestation period on the planet belongs to this shark, very attractive or unattractive, depending on your viewpoint, shark species. And finally, I mentioned them before, but the greenish shark has recently, within the last few years, been proposed to be the oldest living vertebrate. Estimates of age that were deduced from the eye lenses, typically sharks are aged using their vertebrae, but the greenish sharks and typically cold water species and deep water species, their vertebrae don't calcify very well. So they can't use the vertebrae to age them. They tried a new technique using eye lenses, and they estimated that they could live somewhere between 200 to 500 years of age, which would make them the oldest vertebrate on the planet. I think 500 might be a bit of a stretch, but I'd easily believe somewhere from 200 to 300. Sharks are fish. I mentioned the whale shark and is the largest fish in the ocean or on the planet, but they are different to teleosts, which are often referred to as bony fish. And based on the name, as you can maybe guess, that's largely comes down to their skeleton. Sharks have an entirely cartilaginous skeleton. The only truly calcified material in the shark skeletal structure is actually the teeth, which are not obviously a direct part of the skeletal structure in that sense. But the rest of it is a cartilaginous material, giving them the name cartilaginous fish as an alternative to elasmobranchs. Unlike fish, sharks essentially fly through the water rather than swim. And what I mean by this is most teleosts have a specialized organ called a swim bladder. And they use this in the same way a diver can use a BC to maintain buoyancy in the water without actually using propulsion. Sharks don't have this. What they do have to kind of counteract their negative buoyancy, and all sharks are negatively buoyant except for some species that have been known to gulp air and hold it in their stomach. They are negatively buoyant, but they counteract their negative buoyancy with a very, very large liver. It can be up to around a third of their entire body mass can be their liver, and it's very high in lipids, which makes it negatively buoyant and positively buoyant in seawater. So that counteracts their negative buoyancy, but they are still, in essence, negatively buoyant even with that large liver. So the way they're able to maintain position and elevation in water is using their fins in the exact same way that an aeroplane uses its wings. The curvature of the fin creates low pressure above the fin and high pressure below that as they move forward through the water, and that in its essence creates lift. Sharks are quite famous in terms of their senses. They have 7 relative to our 5. They have a sense of taste. They have a sense of touch. They have a sense of hearing. They don't have an external ear because it wouldn't be very hydrodynamically appropriate given the increased density of seawater relative to air. But their inner ear system is quite similar to ours, but they're mainly interested in kind of lower, bassier sounds, things like struggling fish, etc. Sharks' sense of sight varies dramatically between species. If sharks are living in shallow, clear water in, say, the tropics, they often have a very good sense of sight and use this a lot during predation. However, if they're either deep sea sharks or sharks that are living in murky water, they'll often rely on their other senses more heavily. Their sense of smell is quite famous. There's a lot of myths around sharks or facts around sharks you hear being, say, a shark can smell a drop of blood in an Olympic swimming pool, or a shark can smell blood from 2-3 miles away. Their sense of smell is good. It's not particularly better than most other fish. But what's different is they have very large brains relative to other fish, and their brains are largely geared towards their sensory systems, and they can interpret their sensory cues a little better. So some shark species in particular who rely very heavily on olfaction for predation have a very good relationship between their sensory organs and their brain in order to try and locate scent sources. But in terms of smelling blood from a couple of miles away, they'd have to be down current, of course. They can't reach out and smell. The blood would have to be more mixed with the water from an up current source for them to be able to do that. The sixth sense is the lateral line system, which is common to most fish. This runs around the head, around the not exactly central, but semi-central line of the body and up the upper caudal fin. And this is a pressure sensing system. This, for some fish, is used heavily in schooling. So when you see things like a bait ball that are moving around as a single unit, they're able to do that in part by using their lateral line system to detect the distance of the fish next to them in that bait ball. And as that fish moves, they can sense that without having to visually observe it and move as a single unit. Sharks don't really school that tightly. Some sharks don't school at all. And the ones that do, things like scalloped hammerheads, they're not schooling that tightly where they're needing to move as a single unit. But they can use it for things like predation. So, for example, if a viable prey item was moving next to them away from their mouth along the side of their body, they could know exactly where that item was, and that helps them in obtaining that item when they turn to strike it. And one of the more exceptional senses that the sharks do have, and that is incredibly sensitive, is the electromagnetic reception. They have a series of pores around their snout and their head called the Empalia lorenzini. And these are very high resistance electrical pores. They're almost like if you turn a shark over and look at the underside of its nose, if it's a light colored shark, there's a pattern of these things. It looks like someone sprinkled pepper on their nose, but in a very deliberate pattern. And behind this electrically high resistance pore is a tube that is filled with a potassium-rich jelly. And on the other end of that, where the nerve is, there's another high electrical resistance. And along the tube, there's millions and millions of tiny, tiny little hairs. And as electrical fields pass up through the Empalia lorenzini, they displace these hairs. And the amount they displace these hairs can tell the shark how strong the electrical field is, or the electrical pulse is. And the direction where they're being received, more or less on the direction of the tubes going up through their head and into their sensory system, tells them the direction that the electrical field is coming from. And they are believed to use this for navigation, particularly in the open ocean where they won't have any visual cues. We have many shark species that will cross oceans, and we believe they're using the Earth's electromagnetic map to navigate. But they also use this for predation. This is particularly useful either in low visibility situations, or when potential prey items bury themselves in the sediment. They can locate them using their other senses like smell, but when they get very close, they can revert to this electromagnetic reception to locate them using the electrical pulses being created by the movement of their muscles, or even the beating of their heart of that particular muscle. And here's a very quick demonstration to explain what's going on here. This is in Hawaii. This is a baby scallop hammerhead shark. These 4 visually identical rings are visual cues, and in the center there is odor source, which is probably fish based. The shark comes to the area. Oh, I seem to be experiencing a problem. You guys, I don't know if anyone can turn on. My screen's gone blank, my primary screen. I don't know if that's my screen or the...
I can still see your PowerPoint and the video is still moving. So
okay. So I'm not exactly sure what's going on here. My primary screen's gone blank. It could be a connection issue. I can revert to the secondary screen and still go through the presentation to save time.
Oh, that's fine. You can.
Okay, so and essentially what's going on there is the The one where the shark is striking at, that's where the electrical pulse is turned on, and they could turn it on from different locations. And the shark, wherever that pulse was turned on, that's where the shark would strike at, as they were able to detect that. I might be having a more significant problem as
yeah, now it's I'm seeing shark fin soup now instead of
okay.
I've
got some kind of issue here. I can't see anything on one screen, and my other screen is frozen, which is fantastic. Give me a second. I may have to, and escape doesn't seem to be working. I've got an idea. Hello?
Yep, still here.
Okay, I've got complete darkness now. I don't know how to resolve it. I think it might be a computer issue. Sorry about this, guys. I've got
no, that's fine. If you need
a computer, and it is giving me problems since the day I've had it. I don't know what the best resolution for this is, because I can't remember completely the slides, and I don't think I could do this blind. I may have to restart my computer. Will you guys be able to hang tight for a second while I do this? I'm sorry.
Yeah, we can do that. That's fine. We'll keep the Zoom meeting going so you can leave and then rejoin, because you still have access to the link. So
okay, great. I'm sorry about this. It would maybe take me a couple of minutes. I've just got complete.
You're fine.
I can hear you, but that's it. Okay.
Zoom this and then go ahead and share your screen, and we can figure out too then if it needs to.
I think you need to make me host again.
Oh, yep. Let's see.
This is the joys of virtual presentations.
All right, try that now.
Okay. What do I want to do? I think this cool. I was going to do this. Right. Okay, you guys see me?
Yep, we see you, and then we also see the presentation, not in presenter view, so you're all good.
Okay, great. So I'm going to skip over some of the biology and ecology here. There's three main forms of reproduction. They have a case-selected life history, which the basic takeaway is it makes them highly susceptible to predation, as they don't have the recovery potential of most as a lot of other fish species do, even when exploitation ceases. Big demand for shark fin soup causes a lot of sharks to be killed annually, estimated around 100 million individuals per year, which is around 35% higher than what would be ever considered sustainable. This has led to around a quarter. This is a 2014 paper, so it's probably a little bit out of date now. But at the time, around a quarter of all elasmorank species were threatened with the risk of extinction based on overexploitation. Sharks are important to our ecosystems. This is a very, very simplified idea of top-down effects, but they do, as a kind of upper-level predator, have a disproportionate influence over marine ecosystems, and removal of sharks can have quite catastrophic disrupting effects to marine ecosystems. So for healthy ecosystems, we do want to sustain sharks, not only sharks, but they are an important part of that. But they do have a direct economic value beyond exploitation. The example I'm going to use is the Bahamas. It's estimated that shark dive tourism in the Bahamas is worth to their economy around $113 million alone, whereas a dead shark one time at market would be worth around $300, primarily for its fins. And it's estimated that each shark kept alive each year generates around $15,000 for the Bahamian economy. So you can put an economic value on healthy shark populations for an island nation. Oh my God, I'm going to freeze the game.
Because we see the economic value. There you go. Now the slide.
Okay. Yeah, I think it's working again. So I'm going to give you an example now. We need research. We have a problem. We've got declining shark populations for many species. Not all species. Some are just fine. There's over 500 species of shark, and some are doing just fine, but a lot of them are being exploited and populations are declining. Now, I'm going to give you a very succinct case study focused on this species. As you can see circled here, it's not that apparent, the lemon shark. And the reason the lemon shark is a good study species is because it's from the group Carchariniforms, which is the largest group of sharks. It is around 235 of the 500 known species. So broadly, things we find out about lemon sharks can be applied to lots of different other species. Whereas if we looked at something like a wobbegong shark up in the upper right corner there, that data isn't going to be so applicable to other species. And not only for that reason, but lemon sharks are one of the most intensively or comprehensively studied shark species on the planet. This is largely due to the work of Dr. Gruber and his colleagues in Bimini in the Bahamas, where I undertook my undergraduate and PhD research. This is somewhere where Dr. Sorensen has been many times with groups from your university. But largely the work of Gruber and the Shark Club was focused on the juvenile individuals, to a lesser extent, the subadults, the kind of teenage size sharks. What we knew about the adult lemon sharks was largely extrapolated from the breeding biology and the genetics of the juveniles contained within the nurseries around the island. And that's partly because we hadn't had a good, reliable population to study. And the adult phase of case-selected species is arguably the most important, because it probably encompasses the most cryptic and the most significant behaviors in terms of their ecology and their protection, and it includes the reproductive phase. This changed in 2005 when we were sent this video by a local sports diver off the coast of Jupiter, Florida. Jupiter is a bit north of Palm Beach, if you're familiar with the Florida area. And he sent us this video of adult individuals that were aggregating in a previously undocumented behavior. To give you some perspective, the sharks kind of in the center of this that are swimming, they're probably about 3 meters in length. So these are large, lemon sharks only get to about 3.5 meters total length. That's about 10, 11 feet, I think, would be the upper limit of a lemon shark, and that would be a really big one. But these are big individuals, and they're aggregating en masse. We started studying them because it was the first time we had a reliable population, and our study began in 2007. However, as we kind of established this study with the primary goals of testing the theories of the adult phase that have been extrapolated from the genetics of the juvenile, a serious conservation issue threatened these aggregations. This really came about in 2008 with the prohibition of the sandbar shark. The sandbar shark was the bread and butter of the small-scale US shore-based shark fishermen, which is probably the largest shark fishery in the US. A legal fishery, but given the limits and the policy and management practices put upon them, sharks had to be landed whole. They couldn't fin the sharks as of the early 2000s, so they had to land the sharks whole, and they were typically operating out of small vessels, which are shore-based in short trips from shore. By targeting sandbars, the sandbar sharks themselves are pretty large, but they're quite stubby. They're not very long. And their fins are very big. You can see these big dorsal fins and these big pectoral fins relative to the length of their body. This meant by targeting sandbar sharks, they could fit more on their small boats and get maximum value from those fins, which are much more valuable than the flesh of the shark itself, the meat of the shark. So they were prohibited in 2008, which means that commercial shark fishermen had to turn their attention elsewhere. Now, next up was the lemon shark. The lemon shark are quite considerably bigger than sandbar sharks, and they're a lot longer. They're not as stout, but they're a lot longer. But what they do have that was a benefit to the fishermen, or attractive to the fishermen, they have these very long pectoral fins like the sandbar shark. Their first dorsal fin isn't as big as the sandbar shark relative to their size, but one of the defining features of the lemon shark is they have this very large, almost same size second dorsal fin. If you look at the dorsal fin, second dorsal fin on a sandbar shark, it's almost non-existent. So essentially, by catching lemon sharks, they're getting 2 dorsal fins for the price of 1, or the effort of 1, and this was very attractive to them. But in Jupiter, Florida, where they were being largely targeted, the aggregating behavior had the potential to dramatically increase fishing pressure on the population of lemon sharks, as we know from other species, such as those studied by Dr. Waterhouse and Nassau Gruber. Their population decline was largely caused by targeting of spawning aggregations. Now, we didn't think these were spawning aggregations. Sharks don't spawn, but this aggregating behavior and the knowledge of it by the commercial fishermen was potentially very detrimental to these populations that were also believed to be seasonal, mainly occurring in the winter months. So we came up with the following questions. When are the sharks aggregating? They've been reported by the local divers to be aggregating in the winter months. Was it a case that they were staying in Florida during this time, but just moving elsewhere where divers weren't frequenting, or were they going somewhere else? And how did that relate to the population or the catchment area, say, of these aggregations? The second question in relation to that is, where are these sharks coming from? What are we talking about in terms of the catchment area for these aggregations? And the third question was, why are they aggregating Jupiter? Is there something specific about this location that is causing them to aggregate? Or is this a common behavior that is probably predominant throughout their range, and therefore Jupiter is not particularly special and not of particular concern? We were able to catch sharks. We were introduced to the next commercial shark fisherman. We originally tried catching them with rod and reel and were pretty unsuccessful, but he introduced us to a commercial technique called drift fishing, where you fish single baits on single floating buoys, and they drift in upstream with the Gulf Stream current. And when a shark is captured, the shark pulls the buoy out of line, and you can see that it's caught, and you put them up to the side of the boat and work them up. We use the technique to answer these questions. Was like the primary technique was something called acoustic telemetry. If you're not familiar with this, it's a system by where you implant acoustically coded transmitters into the body cavity of the shark. You can attach them externally, but it's much more efficient if you implant them internally. They have a 10-year battery life, so we want to get them most out of that time. And if you put them externally, they will either be bitten off by associated fish, thinking they're parasites, or the sharks are quite clever at scraping them off. So we put them into the body cavity, and they'll stay there for the rest of the shark's life, and they'll transmit codes for 10 years. These codes are then detected by receivers which are established around your study area. And in addition, in collaboration with other scientists throughout the US who are using this technology and throughout the world, we can track the larger scale movements of these individuals as they'll be detected by everyone else's receivers. Here's a picture of a receiver mooring, and this was our setup. We had a core area of 18 receivers that was established around the Jupiter area, and our furthest north receiver in our personal array was up in Hope Sound, and our furthest south was just south of Delray Beach, this Delray Beach Inlet. We tagged 100 sharks, 50 males and 50 females with 10-year battery life acoustic tags. And we collaborated locally with the Florida Fish and Wildlife Commission, who had a lot of receivers out, and they were tracking snook and snapper with them, but they are all compatible, so we were able to get data from them. And this is a first result. This just shows the number of detections essentially on each receiver. And what we found quite quickly was, despite tagging sharks along this whole stretch, they really did tend to congregate around the Jupiter area. So it did appear that there was something specific about the Jupiter area that was causing them to aggregate there. Our first question, when they're aggregating, they've been reported to be aggregating or present in Florida in these aggregations during the winter months. This was found to be true with the data we had. The blue bars are the number of detections, total number of detections per day. The red squiggly line is the water temperature that we were able to document using temperature profilers at the receiver sites. And we found that there was a strong correlation with water temperature. When water temperatures locally would drop below around 24 degrees centigrade, the sharks would show up. This was typically around mid-December. And when they consistently rise above 24 degrees centigrade, most of the sharks would disappear. You can see there's a few individuals that did remain in the area, but the bulk of the activity was around the winter months. So that was the answer to our first question. The next question was, where are the sharks coming from? Well, tapping into this extremely widespread deployment of these acoustic receivers and the various data networks and array groups that exist, we're able to determine that in the summer months, some stay in Florida, a few individuals, but the rest of them spread out across a wide area of the US Eastern Seaboard. In fact, everywhere from Jupiter, where we were tagging them, all the way up to Cape Hatteras. So they'd all travel north, and they'd basically stop off along the way, and individuals would stay at various locations along the way. So this answered where the sharks were coming from. And what this helped us determine is that these aggregations do represent the entire concentration of the entire East Coast population of this species. So this is a seasonal concentration, therefore targeting of these aggregations could be very detrimental, not only to the local lemon shark populations, but annually to the entire Eastern Seaboard population of lemon sharks. The third question was, why are they aggregating Jupiter? Well, we believe it comes down to bathymetry. Here is a model that's Florida, the lighter part, and then this is a model of the continental shelf. What we see is as you travel down the coast from north to south, the continental shelf becomes extremely compressed around the Jupiter area. And what this causes, this is a satellite sea surface temperature image for the month of February of 2009 when we were tracking the sharks. In the summer months, the warm waters push north and the sharks can follow those warm waters north and spread out. In the winter months, the waters drop below their thermal tolerance and it forces them south, probably along the coast. They're a coastal species and they congregate south. And once you get to the Jupiter area, it's the first time they encounter the warm waters of the Gulf Stream coming out from the Gulf of Mexico. Therefore, they have no further necessity to travel south. So this likely congregates them in this area of suitable water temperatures. And we believe at this time they congregate due to both A, social interactions, and B, structure. All these aggregations were documented around either artificial or very structured natural reef areas. So that was the answer to our final question. Based on this data, we submitted a report to the Florida Fish and Wildlife Commission, who oversee all the shark management for Florida state waters. And in this report, we demonstrated that from our data that just to give you a timeline of this report, the aggregation was discovered in 2005. We were able to get funded and initiate a study by 2007. By 2009, we felt we had sufficient data to address these questions and submitted a report to FWC. And within this report, we were able to demonstrate that the seasonal high concentration in Jupiter, Florida represented a concentration of the entire eastern seaboard population. Obviously, aggregating behavior makes them highly susceptible during their time in Florida to population decline from unsustainable fishing pressures. They were being targeted as an alternative to sandbar sharks. And as such, we proposed that lemon sharks required increased protection during their time in the winter months in Florida waters. And specifically, we proposed the addition of the lemon shark to the Florida prohibited species list. Now, amazingly, in 2010, Florida Fish and Wildlife agreed. It wasn't just a simple, they looked at the report and said, yes, we agree. They proposed a rule change, a law change. It was taken to several public hearings and eventually passed into law in March 2010, making it illegal to target and kill lemon sharks anywhere in Florida state waters. Not making them completely protected because they can still be targeted in international waters, but definitely increasing the protection for this vulnerable species during their time in Florida. So let's see how we're doing with time. I'm going to breeze through this as that interruption definitely cut in. So that's one example. There's lots of other different species of sharks. One of the places sharks are very plentiful is in the Bahamas. And SHED's research programs, until recently, and I can't get into this because we just don't have time, there's been political issues in the Bahamas that have restricted our ability to get permits to do research. But for many years, decades even, SHED has been doing research in the Bahamas. Focus research really started back in the 90s with my boss, Dr. Chuck Knapp, the Vice President of Conservation Research. And he established an endemic iguana research project in the Bahamas. But SHED has been collecting fish for public display in the Bahamas since the 1940s. So we have a very long-standing relationship with this country and a very long-standing connection to these ecosystems, which is why our research focus had been based here. And during that time, we built good relationships with relevant in-country partners, particularly the Department of Marine Resources, who oversee all the fisheries for the region, and also the Bahamas National Trust, who oversee all the national parks, both terrestrial and marine. And working with them, we developed our research programs, including the shark research program. We're able to conduct research in this area as SHED, in addition to its facility here in Chicago. We have a very large, well, relatively large research vessel. It's an 80-foot aluminium hull research vessel called the Coral Reef 2. It's originally built for collection purposes back in the 1980s, but has largely been converted into a primary research vessel. So this allows us to travel out to the Bahamas, not from Chicago, the boat's based down in Miami, and conduct research out in those areas. The marine program itself is an ecosystem approach program focused on the conservation of economically important species. We used to have 4 pillars. We currently have 3. as Dr. Lynn Waterhouse was overseeing our grouper program, and she has since moved to your organization, the University of Minnesota. We haven't refilled that position as of yet, and that largely comes down to the uncertainties we're facing about research in the Bahamas, but we hope to do that. But we have different species. We have sharks at the top of the ecosystem, coral at the bottom of the ecosystem. Those are not so economically important from an exploitation standpoint, but economically important from a tourism standpoint, and also helping to create healthy ecosystems that can sustain, help to sustain sustainable fisheries of exploited species like the grouper and this other species here, the conch. We also focus on lobster, very important economical exploited species in the Bahamas. And the long-term hope is that we can use these different trophic level studies to understand top-down and bottom-up ecosystem process in the long term. All our research being applied with real-world conservation outcomes. From a shark perspective, sharks are very plentiful. There's lots of biodiversity of shark species, probably the most biodiverse and abundant shark populations in the Northwest Atlantic. And this is because in 2011, the Bahamas designated the entire national waters, the entire extent of their national waters, which is around 250,000 square miles, as a shark sanctuary. And this made it illegal to kill any shark species for exploitation anywhere in the Bahamas, which is fantastic. You might think to yourself, why would we focus on a research program where sharks are already technically fully protected? Well, it is a policy, and there has been opposition to this policy from certain sectors, both in the government and the private sector. And there's been threats to overturn this policy. So a big part of our mission is to continually demonstrate, this our second goal, the near and far benefits of the Bahamas Shark Sanctuary. And one of the ways we do that is for our first goal, which is describing the abundance and biodiversity of shark and ray species in both shallow and deep water ecosystems. And that's largely descriptive, so we can help assess the state of different shark species, but that also helps us to monitor temporal trends and look at the effectiveness of the Bahamas Shark Sanctuary in protecting and sustaining these abundant and biodiverse shark populations. We use different techniques to do this. For the abundance and biodiversity, specifically, we use both shallow and deep water long lines. We have a winch, a deep water winch on the boat. This allows us to sample down to around 2 kilometers, around 2,000 meters. I don't know what that is in feet, maybe 6,000 feet. And we catch lots of weird and wonderful creatures down there. This is the very large eye of a gulper shark, a deep sea gulper shark. Here's a big tiger shark we brought up. This is a more typically shallow water species, a Caribbean reef shark. We also use acoustic telemetry. This helps us to understand how individuals that reside in the Bahamas are connected both within the Bahamas and to the surrounding area, and that gets at the far benefits of the Bahamas Shark Sanctuary. What are the spillover effects from this effective management? We also use a technique that is linked to the University of Minnesota called BRUVs, baited remote underwater video surveys. And here we can use these surveys. They're very simple. We put down a frame with a bait cage, a kilogram of bait, and a GoPro. And then we basically, very, very high level, we count the total number of sharks we see in any one frame at a time. And this gives us a relative abundance of each species and also the biodiversity for a given location. The BRUV research is also contributed to a global study on shark tropical and subtropical reef-associated shark populations called Global Finprint. It involves, there's a little summary on the side, 121 different scientists, 731 volunteers, 58 countries in the tropics and subtropics have been surveyed. And it resulted in this large paper, this large nature paper, giving an overview of global status and conservation potential of reef sharks. And SHED not only contributed footage to this, but we also contributed analysis. Our volunteer department helped, in addition to our team lab, helped us analyze over 1,500 hours of the 15,000 hours included in this study. So a huge proportion of the analyzed videos were analyzed by SHED volunteers and researchers, and including one woman, Betty, who's still involved with the program, who personally analyzed over 800 hours on her own, which is really incredible. This work continues in collaboration with the University of Minnesota. Dr. Lynn Waterhouse and her master student Brianna, who I think is on this call, are helping coordinate the continued analysis of this. And Betty is still, to my knowledge, analyzing videos. So this is ongoing work that is collaborative, both with Shed, Global Finprint, and the University of Minnesota. Just to summarize, I work at an aquarium. This is not where I thought I'd end up working. I thought I'd end up with some kind of assistant professorship at a university in the academic realm. It's been an education. It is an emerging area of employment for marine scientists. More and more aquariums are recognizing the necessity to have dedicated conservation research programs as part of their repertoire, and often as a forward-facing part of their repertoire. They're unique places to work. We have animal care and science divisions. We have collaborative people within that. Big expertise in animal husbandry, which helps us with our research, and also animal health with our veterinarian departments that can help us. We have a large audience, both in person. We have around 2 million visitors a year, and a large online social media audience. And just from our website, we have an education department that can help us disseminate our results into curriculum and our findings. We have a big policy and advocacy department, and also a big communications team, which help elevate the scope and reach of our findings. So again, apologies for the interruption and rushing through that last section, but I'm very happy to have given this presentation, and happy to take any questions or receive any comments or reflections. Thank you.
Thank you so much. That was a great talk, and we appreciate all that cool stuff that you had on the array of sharks. And if anyone has any questions, feel free, you can unmute yourself, or you can put them in the chat and I can read them off. So you have one from Jaron, and he said, is there any evidence to suggest that climate change will alter the aggregating behavior of lemon sharks in Florida?
Yeah, it's possible. We'd be talking pretty long term that would occur, but what we did find, our kind of primary finding from the study and the resultant paper was a publication, was that temperature drives their behavior, both in terms of their migrations, their annual migrations, and the timing of their annual migrations, north and south, but also locally, we're able to demonstrate that very fine scale local movements were in response to temperature changes. Therefore, with continually warming oceans and the timing changing of the temperature profiles of the oceans, it will probably increase the ability of the sharks to reside further north. So at some point far in the future, it could be that they don't need to come as far as Jupiter, and it will redistribute where they aggregate. And additionally, it could also increase their most northerly range. It seems like Cape Hatteras was the most northerly point they would travel. That's probably because you get the Arctic recirculation gyre coming in north of that point, and that's typically colder water. So when they round the point Cape Hatteras, it's probably a bit too cold for them. But as temperatures increase, it could increase their summertime distribution further north, as well as potentially displacing where they aggregate. So it's possible, but we'd probably be talking a little ways down the line.
Great, thank you. Any other questions? All right, you have them from Danny. What's the extent of collaboration for acoustic telemetry monitoring across countries, organizations, etc.? If you have a shark that is recorded on someone else's monitor, do they have to send you the reading, or do you have access to other monitors not owned by Shed?
Yeah, it is variable, but it's very extensive. When I first started studying, even during my PhD, we established a group called the Florida Atlantic Coastal Telemetry Array Network, and that was one of the first true networks I'm aware of globally, where all the researchers, including FWC and other people, all the local universities, we got together annually and we coordinated and we shared data. What would typically happen if someone was using the same, prior to this, if someone was using the same equipment and you downloaded your receivers and you had a load of unidentified codes, you would send them to the company that manufactures the equipment. They would look up the IDs, they'd email or call the scientist who owns those tags and ask their permission to put them in contact with you, and then you'd have to share the data. Now, what's more typical, there's many global, and there's some really good papers out there on this, there's many global acoustic telemetry networks where people sign agreements with the network managers they upload all their data and all their tag information, all their downloads, all their tag information to a centralized database. And then the research can either query the database to get all their detections without having to seek the direct permission of the owner of those receivers, because they've already signed off their permission to let people have their own tag data, or they have periodic data pushes where the network managers will send you emails with all your data for the last 6 months, etc. So it's much more efficient now, a much better way to collaborate than it used to be. And through that, we have a paper that's going into review soon that highlights not all, but lots of different examples of unexpected movements and migrations that would not have been revealed in the kind of spatial ecology of a different species or group of animals without these collaborative agreements and without this data sharing.
So you have a couple more in the chat. So one from Claire. What advice do you have for graduate students or early career scientists to grow their science communication skills?
It's not really something that was at the forefront when I was going through undergraduate or either graduate studies. I believe now there's a lot of opportunity, through different schools and even online courses, etc., for that specific. It's a much bigger emerging area. Most of my training really came from just trial and error, but since when I came to Shed, we had a good communication department, and they gave us some specific media training, communication training, etc. So that was very helpful. And I think if you look out at some of these organizations like Shed, they'll have internships with their communications department that will teach. But there's a lot of online courses, and if you look, I imagine University of Minnesota probably has, because science communication there's even courses out there like full undergraduate courses, etc., that are kind of focused on that now, I believe. So a lot more opportunity than when I was coming through.
All right, so it looks like you have one more. This is from Brock. I worked for a number of years on the Pacific coast in Alaska as a fisheries observer and noticed a lot of catch of shark bycatch, especially in trawl vessels, and wondered if you saw similar bycatch impacts in Florida or the UK.
Yeah, bycatch is a big factor for population decline. The primary driver is still targeted fisheries primarily for the shark fin industry. But bycatch is a big issue. Post-release survival on bycatch is very variable between species, and there's a lot of work that got into it. But I'd say globally it accounts for maybe 20 to 30% of shark mortality is related to bycatch. So it is a big issue. Florida, nearshore, not so much. Most of the fisheries operate in nearshore kind of hook and line, so bycatch is minimized. You do get some long lines operating there that have bycatch, but long lines are typically less destructive in terms of post-release mortality if it is true bycatch that's released. But yeah, I can't speak to the UK because they do a lot of trawling out in the UK. But I'd say up in anywhere they're doing mass trawling or commercial level long lines for tuna, swordfish, things like that is where you're gonna have the most bycatch. So for the lemon sharks in Florida, that's probably not the targeting is the primary concern.
Great, thank you. And so with that, we're right at 1, so I want to be mindful of your time as well. Lots of thank yous in the chat. Lynn says thank you. She was on the call. So
thanks, Lynn.
Yeah. So I'm also her grad student. So that's also I'm one of them. So
we miss you in Chicago, Lynn.
I'll let her know. She might have hopped off just a minute ago. But yeah, with that, thank you so much. And we appreciate you taking the time to talk with us. And that was a great talk. So
My
thank
pleasure.
you so much.
Thank you.